from Larenstein. And in this workshop, we're going to all different kinds of restoration approaches for Diadema Antillarum, once-white sea urchins. And when we first started to plan this meeting around 4 months ago, I had never thought that the first session about this meeting would be about Diadema die-offs, but unfortunately, reality caught up with us, and we are experiencing die-offs throughout the Caribbean. So I thought it would be depressing but necessary to start this restoration workshop with the die-offs that are actually happening. I did include one young and healthy settler here to sort of get a little bit of hope in there.
Anything?
Yeah, can you click on the presentation? So what I'm going to talk about in this session is a short history of die-offs that were experienced before, the scale of the current die-off, like how big it is and what islands, some of the characteristics like symptoms, the incubation time that we think that is associated with this lingua mortality, and how lethal it is, like how many actually die from it. We are doing a treatment experiment right now. The people here in Saba are going to see it later on, with some results from that, and what you can do to help. And I have a logo up here, the Diadema Response Network. So as soon as it was clear that this was something bigger than just local die-offs on 1 or 2 islands, a lot of organizations from different islands, a lot of people from different islands joined together in the Diadema Response Network to find out what's actually happening, how big is it, and we're also trying to get, as soon as possible, plenty of samples of healthy and especially sick Diadema to do histopathology to find out what is actually happening. But I want to mention that because part of the knowledge, part of the things that I'm presenting now is also gathered by other organizations. All right, so this, all of you know this, of course, but Diadema Antillarum or Diadema, they are a key herbivore on the Caribbean coral reefs, and they were also the most abundant herbivore before the 1980s. In 1983, a die-off happened, a Caribbean-wide die-off, and it's estimated that around 98% of all the Diadema died off in 1983 and 1984. Why are they so important for coral reefs? They reduce the turf and the microalgae, and they facilitate coral recruitment and coral recruit survival and growth. So they basically make it possible for the reef to recover from other disturbances by grazing away these microalgae that otherwise take over. So the 1983-1984 die-off, before I could just say the die-off, but I guess you have to start using year numbers. Started in Panama, was spread with the currents, and that made people believe that it was waterborne, the disease, and resulted, like I told you, in 98% reduction in densities of Diadema. In the decades that followed, till 2015, it was estimated that the average population density was 12% of the pre-die-off density. So recovery was really slow, but it did actually happen. Other Diadema die-offs that are reported are a much smaller die-off in Panama in 1985, a larger die-off in Florida in 1991 that didn't spread to the rest of the Caribbean. And then in the Canary Islands and Madeira, there was a die-off in 2009. It's not Diadema Antillarum living there, but it's a closely associated species, Diadema africanum. But the symptoms were really similar, so that's why I mentioned it here. Right. So we started working on Diadema at University of Applied Science from Larenstein.
recording with
a lot of local partners and partners from the Netherlands. Wageningen University, Caribbean and Science Institute, AZ University of Applied Science, DCNA Reef, Wortel Product Design, and Civil Conservation Foundation, Stinapa, and Bordeaux Dyson. And the project was partly funded by SIA, part of the Dutch Organization for Scientific Research. We mostly focused on developing new active intervention methods to restore Diadema Antillarum. And in the project so far, it's now been almost 3 years. We developed a new method to culture Diadema. Tom is going to tell you all about that later. We also developed a new settlement collector to collect settlers in the field. And we worked on a new method that we call assisted natural recovery, in which we can actually restore Diadema populations on the reef without having to get them on land first. Of course, it's a great opportunity. So thank you for that, DCNA, to provide us with the opportunity to share all this knowledge with everyone through the restoration workshop. Next slide. Okay, so the 2022 Diadema die-off, and how big is it actually? It started with observations from St. Thomas in the US Virgin Islands they saw Diadema dying early February 2022. And then it seemed to be contained for a couple weeks, and then it started to pop up on different locations on St. Thomas and also St. John's, who's nearby. And then it was quiet for a couple more weeks, and then in March, it started on all different islands. I already mentioned St. John's, but also St. Eustatius and Saba. It was observed on Dominica, on Jamaica, St. Vincent, and later on also St. Martin and St. Bart's. And I also saw a report now, I think, further west in the Caribbean. So we're really not talking about something local. This is really something that's happening already on most of the Caribbean islands, actually. And I'm afraid that there's also many islands that are not mentioned here that just haven't really noticed yet, or maybe they noticed but didn't report it yet. So that's why we also started this possibility with the Diadema Response Network to monitor your Diadema populations and to fill it in on the website from agra.org/sea urchin dial to get a better idea of where this disease is. So we'll tell you a little bit more about that later too. So in most of the islands, it seems to start at the harbor, might be transported from island to island with ballast water or with bilge water. And it seems to be local for a couple of weeks and then it spreads outside the harbor and pops up at different locations that often also have ship traffic. So it can be transported possibly outside the harbor to other places, to other reefs, also with ships. Some characteristics from this new mortality. It's also waterborne because we have Diadema at our land-based nursery that are not in touch with Diadema in the fields, but we do use water from the harbor. Those Diadema also got symptoms. Another characteristic is that infected Diadema die usually 1 or 2 days after the first visible symptoms. So they might be infected much earlier, but as soon as they show symptoms, they die pretty soon. The larvae are not affected. We also have a larvae culture. All the people in Saba will see that later, and we don't see anything there. So of course that's great, but it's also interesting. And also all the sea urchins are not affected. So we have a couple different species, rock-boring urchins and West Indian sea eggs that are living together with the Diadema in our harbor population that got affected, and they seem to do fine. And Marit even pointed to them as the culprit because they want to use their skeletons on top of as protection. All these observations are actually very similar to the observation made in 1983 and also made in Florida in 1991. So that can mean two things. It can mean that it's the same thing happening again, it can also point that diadema are, they have not so many ways to express themselves. And if they got affected with something, they probably won't show it. I mean, there are also a lot of fish and other predators are preying on them. So they won't show that they're sick because that will make them more vulnerable. And then if they're very sick, they start losing spines very quickly. And yeah. So the symptoms might quickly look similar because they don't have so many ways to show that they're sick. Of course, there's also a discussion like, is this really a disease or is it maybe pollution? But we really think it's a disease because most characteristics point towards that. Other sea urchin species are not affected. That would be very strange and it would be pollution because why would they not get affected? Many islands have affected populations. It would be very strange if the certain pollution turns up everywhere. And it also spreads to upstream populations that are not in the direct vicinity of a harbor or another pollution source. So that also points towards the disease. So what are the symptoms? Well, there's a couple, and most of the times they occur, it seems, in a rather strict order, but we also saw sometimes that some symptoms are occurring earlier than expected. But what usually is noticeable first is that the water vascular system stops working. So that means that they cannot use their tube feet anymore. That's what you see first. So they fall off vertical surfaces. At a certain moment, they're just standing in the sand next to a rock where they used to live on, for example. They also sway in the current. I'll show you a video later, but they cannot hold on anymore. They're walking or standing on their spines because they can't use their tube feet anymore. And then you can see it in this picture too. The epidermis, the skin start to deteriorate and start to die off. And then the spines fall off. And the spines also seem to become more brittle. And there's mucus rings or tissue rings around the spines. So all these symptoms are really similar to the symptoms described by Lessius et al. In 1984 and by Clement et al. In 2014 for Diadema africana. So I'll show you these symptoms in a bit more detail. So here you can see deterioration of the water vascular system, so they cannot use their tube feet anymore. So the animal to the left just fell off the vertical side of the tank and cannot upright itself anymore. So it was lying like that for more than an hour. Dalia, would you mind to click on the video? So we could show that. Because here you can clearly see that the tube feet are not working anymore. They're still moving a little bit, although they're mostly swaying the water movements. But they do not adhere anymore. There's no power in it. So they're not unable to properly use it. And the animal is actually standing on the spines and can still move on the spines, and spine reaction is also pretty normal. And then this is a picture made by Kumani. There you see them just drinking in the current basically. And here is a picture I made last week where you can really see them, a video I made last week where you can really see how they sway around in the current, not able to use the tube feet. Apparently pretty healthy, but clearly something wrong if they are going around like this. So here you see an animal standing on its spines. Most of the tube feet are already gone. I don't know what happens to them, but if in a later stage of the disease, you don't see the tube feet at all anymore. So they just seem to fall off and disintegrate. And then we have necrosis of the epidermis, of the skin, and spine loss. And you can see pretty clearly here on this animal that the epidermis that holds the spine together starts to become white and really degrades. So you can see over here, it becomes much smaller also, and then totally just disappears, disintegrates, and then the spines fall off. And there's another small video here. And what you can see here, for this animal, the symptoms of necrosis of the epidermis and spine loss actually are happening before loss of the tube feet. Because here you see like small bamboo stick, and you can see this animal can still hold on to the tank surface pretty well, but all the other symptoms are clear. And later on, this animal also had lots of the tube feet and died. And then what happens is that spines become really brittle, they break off, and you see this mucus rings or tissue rings, we don't really know, but it seems to be just a tissue that starts to disintegrate immediately after the spine breaks off. And you also see it with individuals from the field. And then, of course, you get spine loss, and you get these depressing pictures of deaths in the field. So what is the incubation time of this? How long does it take between infection and when they really show symptoms? It's difficult to say but we have this opportunity to study them in our nursery, in our lab. And we all have semi-closed standalone systems. So we don't have a filter system that continuously pumps up water from the sea, but we do replace water once or twice a week with seawater coming from the sea. So we know more or less where we replaced the water for the last time. And it happened that when we found out that there was a disease in the harbor and that there was a diet in the harbor that we get our water from, we had one system with 50 Diadema in there, it's this system, and it was 5 days since the last water replacement at that moment. And of course, we didn't replace any further water for that system. And then we had a couple systems, smaller systems, with around 60 Diadema in total, and it was 0 days since the last water replacement, because we actually replaced water on that day, and we found out in the afternoon, hey, there's something going on, and we just replaced all this water. So from that moment, we switched both tanks to artificial seawater, and we saw first symptoms in system 2 and 3, 5 days after the last water change. So I would say this is probably 5 days. But then the system that was also that was not replaced on that day, the animal still became sick. It just took longer. That was around 12 days. So we don't really know, but it might have something to do with the amount of exposure. So we replaced water earlier that week when there might already have been something in the harbor water, but not as much. There were not dying urchins yet, so it might not have been as much the exposure. That might have to do with the incubation time. So for now, we'll say it's probably around 5 and 12 days, depending on the exposure. And we have some other indications too, I will show you later, that the amount of exposure to this disease in water really determines how fast animals get sick. So these are some standalone systems here on the right.
Oh, sorry.
Yeah, yes. So how lethal is this new disease? At first, when we first observed it, I immediately had to think back about all the papers in the 80s, like when 93% or sometimes 100% then died. And I told myself it won't be as bad this time, especially not if it's the same thing. Some of them might be immune to this. It might be more like maybe 50% or something. Unfortunately, that was not true. We surveyed it for harbor population. Can you click on the thing?
Sure.
We swim there very regularly in the water very regularly. So I knew that on early March, the 8th of March, there was no dead sea urchins. There was no strange looking sea urchins. So I assumed the population was healthy then, but we didn't do a population estimate. And then we heard a couple of days later, we heard about the first reports of Diadema. That was the day that we also replaced water from the in the tanks. And then there was a lot of swell in the harbor, so there was no visibility, so we were not able to survey the population. And we were able to do that a couple of days later on the 18th of March. And then we counted all the healthy Diadema, healthy looking Diadema, all the sick looking Diadema, and all the dead Diadema. And that was a total of 173. But remember, we had this whole week of swell, so probably a lot of the dead urchins were already removed. So I don't think it's actually 173, it will be more like 250, maybe even 300. But we never counted them, so let's keep it at 173. I counted them again a week later. It didn't make sense anymore to count the dead ones because they were being washed away. We just counted the healthy hooking. I think there were around 10 left, a couple sick ones, and we did it again last Friday. We found, I believe, 5 healthy ones and 1 or 2 sick ones. So it's actually still going on. So we're talking about at least 95, but maybe more to 99% lethality here, at least in this case. So that's really, really bad. And this is, of course, only one site, but Marek, Thomas, and Kimani also surveyed the population on cetaceans that were affected. And I believe they have very similar numbers in terms of the percentage that dies. Okay, so we have all this Diadema in the nursery, and clearly something is happening to them. We saw symptoms in both of our systems, and of course we wanted to do something, at least to try something, not only to save the Diadema in the nursery, although that would be nice, but also to find out more about the disease, because if we know what can cure it, we also know more about what causes it. The pathogen responsible for the die-off in 1983 and 1984 is unknown. It was never identified. The pathogen responsible for the die-off of Diadema africanum in 2009 was most likely a bacterium called Vibrio-Alginolyticus. It's a very common marine bacteria associated with bacterial infections in marine environments. And we had experience with treating other Diadema for bacterial infections with oxytetracycline, oxytet, a broad spectrum antibiotic that works for a lot of different marine bacteria. And both gram-positive and gram-negative, and also targets Vibrio-Alginolyticus. So we thought, okay, this is what we have available. We have some experience with it, and it might cure what's currently happening. So we just gave it a try. And what we did, we used blots of 15 milligram per liter of oxytet, and we followed the recommendations made by Aaron Pilnick and others that were described in the newspaper. And we gave them 3 beds or 4 beds, but we didn't put the antibiotic in the standalone system, because then we would kill off all the good bacteria, and it might end up with a system crash and dead Diadema for other reasons. So we did 2 experiments. Experiment 1, we had 3 tanks with 5 Diadema each as treatment. So we treated them with antibiotic beds, and we had 3 tanks with 5 Diadema as a control, so no treatment. They all were initially exposed, but then we switched to artificial seawater. So then they were not exposed anymore. And they got 3 antibiotic beds every other day, and then we stopped treating them. And then experiment 2, we had another 6 tanks, 3 tanks with 10 Diadema each that received antibiotic beds, and 3 tanks with 10 Diadema each that received that were control. And these tanks, we continuously expose them to the disease, because we continue to replace water from the harbor to see if a bigger exposure would make a difference in how they were doing. And we gave them 4 antibiotic beds of 2 hours. These are some of the first preliminary results. This is experiment 1, so only initial exposure, and we see actually that most of the Diadema are surviving even after 10 days. We have a couple of beds in the control, but that's still only 2 Diadema that died. So difficult to draw any conclusions from that. Those Diadema that died had the same symptoms though, so we're fairly confident that they died of this initial exposure. Then we have the second experiment with more Diadema in each tank and continued exposure, and we see a slight difference between the treatments. The ones with antibiotics are the blue line, and the control is the red line, but both of them are decreasing. So we already know that it doesn't cure all of the animals if you treat them with antibiotics. This might result in a significant difference in the end, but we don't really know yet. And animals are still dying on a daily basis in both treatments, so too soon to tell. We also have some really anecdotal results. We have this very sick animal that was lying on the side, We didn't include it in any of the experiments. We only included like healthy-looking individuals at that moment. But we did give it an antibiotic bath, and 3 days later, it was using it tube feed again and walking on the vertical surface and eating. Really amazing, though we didn't expect that. It almost made me wonder if it was something else we were looking at here, but I cannot imagine. So I've never seen Diadema lying on their side, not being able to come up. So I'm pretty confident it's actually the same thing. And here it seems to work. The antibiotics. So very preliminary conclusions. It is for sure a big difference if they're only exposed once or continuously exposed. One of the things that also might help is that as soon as they die, the Diadema, we remove them from the system, so they don't disintegrate as much as in the field, and that might of course affect other urchins. So that might explain why our numbers are going down more slowly than in the harbor, for example, where it's just a crash of the population. That seems to be it. There might be an exposure threshold before infection takes place, but it might also have to do with a much longer incubation time. And too soon to tell if the antibiotics work. Some positive and some neutral effects, I would say. So I can imagine if you see all this, very depressing, that you think, okay, what can we do? How can we help? Can we gather data, find out what this actually is? And that's why we set up several protocols with the Diadema response network to survey Diadema populations, to sample healthy and sick Diadema. If you have the opportunity to collect future broodstock or like a gene pool for future research, and of course prevent the spread of this disease. So the survey can be really easy. Just count all the healthy-looking, sick-looking, and dead Diadema. If you have the opportunity, it would be great if you can also estimate with use of transects how big the area is. Also look at other sea urchins, like the rock-boring urchins and titmoistus. So far, they don't seem to be affected, but that's only based on a few populations. So we really want to find out more about it. And fill in all your results at agda.org/sea-urchin-diet. And then we get a good idea where this disease pops up, but also which islands are actually surveyed. So it's also very important to report a healthy population. So we also try to collect samples. Of course, not everybody has to do that. So if you are in the opportunity to take samples, store them for histopathology or molecular tissue analysis, then contact the response network at diadema@agro.org, so they can coordinate who's taking samples, and then also we don't have to take samples, unnecessary samples of healthy Diadema, for example. And collect Diadema, I think this is a good thing to do for everyone that has the opportunity, that has the structure in place to keep them in an aquarium or a tank for a longer period of time, because we can conserve the gene pool, and it's really useful for future research, and maybe even for future restocking attempts, although we don't really know. It might not be a good idea because they might be very susceptible to this disease. If you want to collect healthy populations, of course, you need a healthy population pretty far from the sick populations, because we saw earlier that the incubation time might be as much as 12 days. So if it's pretty close to an infected population, they might already be sick, but you might not see it, and then you do a lot of effort, and they will still die in the laboratory. And you need a recirculation system, because if you have a flow-through system, you will just take in water and take in this disease, and they will probably still die. So you need artificial seawater or treated natural seawater, and you need the resources to take care of them for long periods. And further in this workshop, we'll also cover what you need to maintain Diadema and how much you feed them and stuff. So again, we'll hear a bit more about that. But of course, it doesn't make sense if you don't have the manpower and the resources to keep them for, I would say, at least half a year, but maybe closer to a year. So yeah, if you want to do that and you want more information, there's a lot of persons, including our team here, that is involved in Diadema culture and maintaining Diadema. So you can also contact us or the Diadema response network to get more tips and more insight about keeping them healthy. So I want to give a shout out to all the members of the Diadema response network. There's really a great collaboration the last couple of weeks. The subject was, of course, very depressing. But thanks for that. And thank you all for listening. And if you have any questions, you can ask them right now to me or later on, especially questions about survey samples or root concentration, contact diadema@outloud.org. Francesca.
I'm not sure if you mentioned that. So would you suggest if you see sea urchins to actually remove a lot of water to avoid or prevent the contagious that you can get from the disintegration? You said that, you know, the disintegration would be actually an extra.
Yeah. That's a very good question. So the question is for the people listening through Zoom, if you see a sick diadema, should you like take it out of the water to prevent further spread? That's your question, right?
Yeah.
Yeah, exactly. And I think that's a good idea if you're still in an initial phase of disease. You could try at least. What we've seen in the harbor is that within a couple of days, you see them dying everywhere, and even if you mutate them, they disintegrate, and it's basically too late. So honestly, I don't think it's going to make much of a difference. But if it's really the first of the population, yeah, why not try?
Or maybe if you don't have like sometimes have random, very spread apart sea urchins, not very close to each other. Maybe in that case.
Especially, yeah, you're right. So if the population is less dense, then that might be a useful strategy. Yeah. Any other questions?
Let's say the antibiotics end up working or just working like a little bit. Have you thought about applications to do that in the field? How to give them baths or inject them? Or...
Yeah, it's a very good question. So say the antibiotics or any other treatment works, can we use it in the field? And of course, I've thought about that a lot actually. And to be honest, so far I don't think we can save diadema on a large scale from this. I just don't see how it's feasible. Also, a single bat won't be enough. Inject them will probably kill them. So yeah, very difficult, I think. But I hope somebody else disagrees and finds a solution. But I don't think we can save infected population on a large scale. Right.
But you guys started like selective breeding, and you see like there's only like 3 live urchins in the harbor. Do you feel like those urchins are stronger or can handle the disease?
Yeah, so that's a very good comment. Marijn says like maybe the survivors are immune or stronger, and then of course, if that's the case, it would make sense to continue breeding with them, and then we wouldn't even use the collected gene pool that I described earlier, at least not for future restocking attempts. They would still be very interesting to look at the gene structure. But we don't know yet. We don't know if they are immune or just lucky, or but definitely look into that.
Maybe too early, but do you know if you have a sea urchin that's recovered, can they get reinfected?
Can it get reinfected?
Yeah, so let's say one of the ones that you treated in the bath, can they, and then you put them out, you would expose them again to the pathogen, can they get reinfected, or is it like a COVID vaccine?
Yeah, exactly. Yeah, so that's a very good question here. Like, can recovered animals, individuals, can they get reinfected? We also don't know yet. So far, most of the ones we saw with symptoms, they just die, and they die so quickly that they're already like on their way dying. The only exception is the picture I showed you earlier with that one that lost control of the tube feed and is now healthy again. Yeah, I'm not sure if I want to give that a try.
Are you seeing mostly only the adults being infected, or also like for example the juveniles?
Yeah, so the question here is, are only the adults affected or also the juveniles? In our lab, it's mostly juveniles that we have, a few like subadults, and the juveniles get infected too. The only thing that the larvae don't get affected for sure. We don't know about the settlers, but they're very similar to the adult urchin, so they probably get affected.
Sorry, Owen, it's about to go into the next session.
Sure, yeah.
Quickly. Maybe, because we may have questions from the Zoom as well.
Yep.
We can take a look at the chat and then save the questions until last before lunch.
Yep.
Does that sound like an idea?
Yep.
Yeah, so we have a lot of questions. To those on Zoom, we'll get to these questions after the next session. Thanks.